Hi, everyone, and thank you so much for joining us today. So before we get started, I do want to share with everyone that now that we are officially launching, we've updated our company's name. You may have previously heard of us as Numerix, but Numerix is now Countable Labs. This reflects our vision to create tools that bring true, direct, single molecule counting to your labs. So I'm very excited today to introduce my colleague Eileen Chum, who's here to introduce Countable PCR. She hails from the NGS space. There, she specialized in both assay and bioinformatics development in the realm of single-cell sequencing, molecular barcoding, and multinomic products at both cellular research and BD. She also led technology development at the leading liquid biopsy firm, Guardant Health, overseeing thousands of clinical NGS data sets of oncology patients. In 2019, she joined and founded what is now Countable Labs as a founding scientist, where she has been working to make single molecule DNA counting a reality. And today she's here as our VP of Product to share the results of over 5 years of innovation and development and how it's set to change the everyday lives of scientists regularly doing PCR in their workflows. There's a Q&A box at the bottom of your screen. Please feel free to leave questions at any point during the presentation. We'll try to address as many as we can at the end. So I will be back to help with that Q&A session. And for now, I will leave it to you, Aline.
Thanks, Stephanie, for the generous introduction. Today I'm very excited to debut this platform that our team has been working on at Countable Labs and what the founding team has been building for the past 5 years. So I'm really excited to be sharing our platform today and really explain how we approach this problem and how we solve the current problems in the PCR space, and obviously show some data that we have collected. First, I do want to kind of talk about what drove, the founding team into building this new platform, right? So when we started at Countable Labs, coming from the NGS space, really, there's a lot of innovation in that space. And then when we looked into the PCR space, what we immediately see is that there is the technology development part of the PCR space is definitely more stagnant. And as we talk to scientists and users of PCR and NGS, what we've learned immediately that PCR tools are used as a workhorse for their workflow, but it is becoming apparent that the current tools available don't really meet the growing needs. And for today, I want to focus on a particular sort of segment of the PCR space, which is in the cell and gene therapy space on viral vector characterization, on how the current tools really lag behind the growing need for developing safe and effective products. So just to kind of summarize, as we talk to a lot of these users, right, in this space where the current tools that are available to them are falling short, they include, say, they have to run different platforms, whether it's qPCR, dPCR, NGS, ultracentrifugation, ELISA, or a combination of all of these to really understand their viral vector batch. So imagine if you're doing a characterization piece using these platforms, you have to correlate all these different outputs to get sort of an assessment of your batch can be very difficult. And two is a lot of these tools require a lot of material. In some cases, we've heard it could use up to 50% of a particular manufacturing batch. So that equates to requiring more cost and effort to really characterize this material and make more of these material. And three is even with those tools that are available in this space, the scientists are suffering from high variability of their quantitation, whether it is between operators or different days and whatnot. So these are really the troubling pieces of how the available tools fall short. And where Countable PCR really delivers is, when we sort of develop this platform, is really to fundamentally provide a very reliable way for scientists to analyze their gene targets, right? So our technology, which I'll show you a bit about later in the slide, is to showcase our absolute counting technology, how we deliver low variability. And two is with our counting technology, what is really cool is that you can now think about new types of assays that you can develop on this platform. So things that are very, very difficult to implement or near impossible to do with legacy PCR platforms, you can do it with Countable PCR. And three is, even though that we can do very like new types of assays, I want to emphasize that these assay development efforts on Countable PCR system is also very straightforward. So not only will it accelerate product development effort, it also makes analytical workflow much more straightforward as well. So before I get into sort of the nitty gritty, I just want to showcase, the feedback from one of our early access customers. This is an email that they sent to us when after we placed our system into their lab, and very soon they have already sent us this email about how they've already moved all of their runs to Countable PCR instrument, and that they don't plan to use the old instrument anymore unless they have to reproduce data from previous runs, right? So that was very encouraging because it was very immediately noticeable. And just want to summarize the pieces that they also talked about is that immediately you can sense that the platform that we have is more sensitive than the platforms they've used before. And two, with the capabilities of Countable PCR, there's an opportunity to replace NGS for some characterization studies. And three is that we can, they've been leveraging this novel multiplex strategy we have to do assay development. So they, instead of having just at that time, there are 5 assays that they run repeatedly, they can actually look at their vector from different angles and develop new assays very quickly with our multiplex strategy. So, I want to give you a snippet of like how Countable PCR transforms your workflow. So just to showcase, right, what are the limitations of current PCR systems, right? So here is some data where we generated using the same DNA dilution samples. On the left is qPCR. And for those, I'm sure you guys are familiar with qPCR as it's such a workhorse tool. But one thing about qPCR is if you don't have standard curve, you don't have the accuracy. You can see the data to the expected line is not accurate. For digital PCR, it works until a certain range where it is either near its upper range, where in that sense you start getting these increasing amount of Poisson error, which adds to the variability to your workflow. And to the point that if you add so much DNA that is out of its range, it just saturates and you don't get a number that's accurate at all. But on the other hand, using the same DNA samples, I wanted to showcase what Countable PCR can deliver. It's a very simple platform where you can basically get the data that is not only high dynamic range, but across the entire counting range, very high precision. I want to emphasize this is 4 replicates tiled on top of each other. Yep. So how does the platform work, right? So I'm excited to share with you how Countable PCR fundamentally sort of works differently than the legacy platforms that you're used to. So just wanted to kind of highlight, right, how the PCR tools you may be familiar with now are essentially estimating what is the true quantification or the true number of targets you have in a tube, whether it is just measuring the amplification fluorescence signal or the ability to convert this into a digitized signal to estimate counts. How we do quantification in Countable PCR is essentially counting, right? So if you imagine you have this many molecules in the tube, what we deliver is essentially a tally or a count of how many molecules in this tube. And how does it work, right? So this platform is actually extremely simple as well. And I showed you, for the earlier schematic being in a PCR tube, and how our workflow works is that it also works in a PCR tube. So to begin with, you set up your PCR mix, and you can see your DNA targets shown in this wiggly worm representation. And it's basically a spin column format that any molecular biologist will be used to. You fit it on top of a PCR strip tube, and then using a very simple centrifugation step, in about 20 minutes, you get a batch of samples where each of these samples now the DNA target molecule is now inside the PCR tube. And the really interesting thing is that we are able to essentially encapsulate these single molecules because we have the ability and the chemistry to isolate these single molecules into around 30 million compartments. So just looking at the number of compartments, it is vastly higher than any platforms are available today. And it really enables the target molecules you have to be isolated by itself. And the cool thing about being able to isolate these molecules by itself is that now you can amplify it in isolation. So you just take the same tube, remove the spin column, and then put it into a thermocycler. And depending on your primers and probes or the assay that you design, these molecules will amplify into a fluorescent signal. Then that you can later use the same tube, put it into our system to perform a simple counting procedure. So our system uses a technology that basically allows you to just scan the tubes directly without pipetting the sample out or like other technologies that are available now. Let me showcase how it works, right? So um, this technique is called light sheet imaging. So essentially what happens is you put the tube into our instrument and it generates these light sheets and it takes a picture of the light sheet that scans through the PCR tube. You can see the outline of the tube that we computationally add, and then you can also see the signal throughout the tube. And in this particular example, this is a 3-color assay where we have either no molecules and a target that has a very high abundance and a target that has, yeah, say medium abundance. And I also want to emphasize that by the Countable PCR platform, this counting technique is done automatically. So you don't need to do manual thresholding, and you just directly get the number of counts of your targets from our reports. And there's also an option to implement an optional machine learning model if you have a positive control sample to really increase your specificity. But by default, the counting is given to you pretty soon. Okay. And one thing I want to highlight, right, is that because of, think about how big the tube is, we have a 50 microliter reaction, 30 million compartments per reaction. We can reach single molecule counting range even when there are 1 million molecules. And on this slide, I just want to show like what it looks like when you load very low number of molecule to very, very high number of molecules at 1 million. So each of these pictures represent the expected input of the molecule that we added from 10 to 1 million. And each picture is basically just one slice across the PCR tube at its fattest region. And you can actually see that, okay, obviously when you add 10 molecules in one slice, you probably can't see that. But as you add more molecules into the system, you start seeing them populating across this light sheet image. And even up to 1 million, you can still count them, and we deliver those counts for you. And in this slide, I'm showing you this sort of one target approach, but actually it works just as well and equally when you have more than one target. So here is an example where this is an oncology example where we are looking at a mutant of the JAK2 gene where we measure the abundance of this rare target versus an abundant, let's just say, control gene or the housekeeping gene, which is the wild type. And essentially, on the right side, I'm showing you the same tube where we image for the hex fluorophore and also for the FAM fluorophore. One is very, very high at near 1 million, and one is in the hundreds. So you can see how in each channel, they also are in different like compartments, but we can deliver those counts in the same reaction. And then the left side, what you actually see is basically the ratio of this mutant we're looking for versus the wild type. So for those who are familiar with sort of like looking at rare molecules You can look at this, limit of detection here. It is as low as 0.004% VAF, variant allele frequency, which is higher than any PCR platform or NGS platform available. So another thing I really want to highlight is the ability not only to be able to count precisely in a high dynamic range, but the notion that we can bring assays, that is really easy to bring up to speed up essentially development time and lower assay costs. So not only really can you deliver great data, but also at a very affordable startup cost as well. So I want to introduce to you guys uh, our proprietary novel universal multiplex chemistry. And for those who I'm sure you guys are very familiar with how PCR works, and this is very simple. Essentially, it is a forward and reverse primer reaction where you have essentially a universal sequence that you tag onto your primer. And after amplification, what you have is essentially these PCR products with the universal sequence attached to it. And of course, you then have these universal probes, which we would provide as a kit. And in these compartments, in these single molecule compartments, now these probes and these products can recognize each other and essentially hybridize and generate a fluorescent signal. So you can do it in one color or more colors because from the kits we can provide a couple of them for you to design these assays. So this is the universal multiplex chemistry that we support. We also support the legacy TaqMan chemistry that you guys are very used to, right? Like the forward and reverse primer approach using a custom hydrolysis probe. I do want to highlight that both chemistries are supported, but the great thing about universal multiplex chemistry, is the fact that it's very easy to use. It's essentially a forward and reverse primer setup, and you don't need like extra probe design expertise. So, and also these primers are much lower cost when you order it from different oligo manufacturers. I'm putting here it's like 10 to 20 bucks per primer, as opposed to hundreds of dollars for a TaqMan probe. So essentially, when you design primers and order these primers for universal multiplexing, you essentially can design, order, and test within the same week. So you can like accelerate a lot of your assay development and really can understand, your gene targets much more and much quicker. Okay. In the next section, I kind of want to kind of bring in, bring back to, our sort of focus today with regards to the cell and gene therapy space where, that current users who are in this space are suffering with high variability. So I want to touch base on how our system allows us to kind of reduce the variability. So there's a lot of aspects, obviously, to variability. So there's your samples, right? But the big part that I want to highlight is that there is the platform that you choose, that you want to be able to choose a platform that reduces the variability as much as possible, so you have the most control of your data, right? So how does Countable PCR really help you reduce the overall variability, right? So first we enable fewer pipetting errors, essentially, right? Think about how if you have a much larger dynamic range, it reduces your dilution steps if you do dilution in your experiments. And also you don't have to, if you blow out a range, you don't have to go repeat your experiments. And two is, we don't use microfluidics, so the PCR volume is much higher. And that means when you don't have to pipette like small number of material that would have higher pipetting error or under or over dilute. You can pipette in a comfortable range where there's low pipetting error. And two is we provide just counts. So there's no Poisson error or needing CT value correction, adding confusion to the data. So overall providing much better precision than before. And three is better data consistency, right? So to capture more attributes from the same platform without needing to correlate data between platforms. So I'll touch base on some aspects that could help you with that piece. But obviously, I'm pretty sure with this type of platform, we can also add more and more applications to it. So just to showcase some data on reducing variability, right? This is a data set, this is a multiplex experiment. I'm just showing you one gene target where we performed, countable PCR across 3 operators. And you can see that between the replicates, the error bar is extremely small. And just to quantify that, between operators, in this experiment, we see a maximum 1.31% variation. And between technical replicates, around 1.1%. So that's really low. So you have more control over other aspects of your sample. And two is the ability to really bring very easy multiplexing into your workflow is actually to be able to get better. For example, if you are doing a titering experiment, if you count multiple targets in one tube in a very easy manner, you can then kind of see which one gives you the better information, right? So I'm just giving you an example here where this is a transgene where it's just a GFP essentially. We use universal multiplex. You can see the primer schemes here, and where we design different targets against different parts of this gene, CMV, GFP, and poly-A. And if you do a titration experiment, this is an experiment we generated, and you can see the counts of the 3 transgenes. So there might be regions of the vector, as we are familiar with, that could be hard to amplify, easy to amplify. So some might be more accurate than others. So you can imagine when you kind of like can do multiple targets at once, you can see which one is more accurate. And 2, another really cool thing is is to be able to now use this type of technique to easily measure full and partial vectors with PCR. And what do I mean by that, right? So I want to reemphasize this design of this multiplex assay. So ideally, in the cell and gene therapy space, you want to create or understand the amount of virus that has the entire transgene, right? So that because that is the one that's working. So the idea of being able to understand this full length or partial length of this transgene can be done very easily using our system. So let me remind you guys that we are a single molecule platform. So you can actually really easily do single linked molecule counting. So what I mean by linked molecule is that if you have a single molecule signal where you have these 3 targets lighting up, you know you have the full transgene. And if you see in that molecule only 1 or 2 of the signal, then you know you don't have the entire piece. So let me show you some data and some imaging. Say we tile these 3 plex together, and I smash it onto an image and zoom in very closely. You can see there's different types of single molecule signal to understand this linkage. So there could be single molecule signal where you only have parts of the gene. You can have only one aspect of the gene, or if for this white, when all 3 colors overlay in this color image, it would be white. And you can see that this molecule is in fact representing the full genome. And essentially, we just count them again. And I want to emphasize this automation, this counting is completely automated. So not only do you count single molecules, you can also just count single linked molecules, and we provide the data for you. And in this example, we basically did a dilution experiment. Instead of just total number of molecules, we dilute the fraction of full transgene and partial transgene. And you can see that for 1-to-1, we're getting around 50%, and probably was pipetting error during the dilution. And as we dilute it more, you actually see corresponding sort of expected ratio of your full transgene. And if you can see these error bars, they're also very tight. So not only can you do linked molecules automatically, you can do this very precisely. So to summarize, I spent some time kind of introducing sort of what Countable PCR platform can do with the focus on viral vector characterization. So just to summarize, one, we can generate reproducible titer quantification across a broad dynamic range. Two is to be able to study linked molecules easily with partial and full genome integrity. Three is we are introducing a new chemistry where it allows low cost and fast turnaround time. So if you have a gene of interest or different parts of the vector you want to look at, you can deliver this data very quickly. And four is very flexible sample input, whether you have very little material or a lot of material, very low volume or high volume, we can accommodate that. And fifth part is overall, we can reduce variability between replicates and users as well because we eliminate a lot of the platform errors that you would see in legacy platforms. So to conclude, I'm really excited to announce that our early access users are experiencing success with the Countable PCR system. So just wanted to summarize what the platform looks like is essentially, an ecosystem of not just the counting imager, but this imager can image 96 samples at once, up to 192 samples per day. You can run 4 channels per run, and all of this data is automated data generated for you, so you don't have to do any thresholding. And two is we also provide reagents, consumables, and PCR mixes to get those uh, reactions going. And the third part is, should you choose the universal multiplex kit, we also provide those kits, and the universal probes are within. So you can basically just take the guesswork on probe design and just put your primers of your targets in and get going very quickly. So with that said, we want to invite everybody here to experience how Countable PCR works for you. So we're looking for more early access users. So we'll be excited to work with you. And with that said, questions welcome.
Thank you so much for that talk, Eileen. We've actually had quite a few questions come in throughout your talk. I do encourage, if you have more questions, to continue leaving them in the Q&A. We'll get to as many as we can, but I would like to remain cognizant of everyone's time. So let's start with the first one that came through, which is, what is the lowest number of molecules you can count in the system?
Yeah, great question, right? When I can count, I would say we've routinely done from 1 million down to 10 to even 0 for our system. And we've had collaborators who are interested at that range as well.
Thank you. And then another question, how many colors are available for multiplexing?
And for our first iteration, we are providing the ability to do 4 colors at once in a very easy manner.
Thanks. And then I think for the imaging pictures, how do you ensure that you don't miss one of those single molecule counts if two of those signals are really close together?
That's a really good question because actually, I only showed the image in like a 2D space and only one image. But how the technology works is that essentially it's a 3D counting tool. So every partition signal is imaged multiple times. And when we looked at the the signal that as a 3D space, so that increases the resolution for you to be able to see whether this is 1 or 2 molecules. In that sense, we deliver you the basically the number of 3D shapes that we see as a count.
Okay. Thank you for that. So another question. What is the minimum and maximum amount of DNA you can load? So I'm not sure which units they're thinking, so maybe let's take us through kind of all the different options
I
there.
see. I think people are interested because it's a high dynamic range platform. It's a I can give maybe different aspects of it. We talked about like 10 molecules or 0 molecules, depending on what your application is, right? So you can go as low as that and up to 1 million molecules, and you'll still be in that single molecule realm. In terms of like, say, mass of DNA, we support up to like 1 microgram of DNA, and 1 microgram is if you put human genomic DNA, it's at the range where already that digital PCR cannot count. And I think it's probably around 500,000. I'm going to take a guess here. So around 1 microgram and down to as low as you want to detect. And I think another aspect, how low or how high the DNA you can load is that our PCR reaction is not done in microfluidics. So by that nomenclature, microfluidics, typically the reaction volume is very small. So the limitation is that you cannot pipette a lot of DNA that if it's dilute, right? So in that sense, having our system being like a normal tube size, now you can load a lot more volume of DNA. So it really accommodates not only highly concentrated samples, but very diluted samples as well. So our PCR reaction is 50 microliters.
Okay, there's sort of a second question that's
Mm.
related to that. What is the upper limit of quantitation? So, you showed the dynamic range, we showed up to 1 million, but, kind of what's the theoretical max on that?
It's a good question. We currently routinely do up to 1 million counts. But if there's like some applications that you're thinking of that, or whoever asked that question, you can chat with us. Um, but 0 to 1 million could provide quantitation counts, yeah, for this iteration.
Awesome. Thank you. I think there are quite a few kind of detailed assay development questions that are probably a much longer discussion. So I will say we can follow up with some of the attendees who have questions about sort of more experimental design and how to
Okay.
apply this to their own work. But let's make sure we haven't missed any more. There's a question a little bit more general about assay development. So when you say optimization is fast, like what do you mean by fast? Is it only that you're saving time with that primer ordering, presumably the universal multiplex, or is this like assay development as well?
Yeah. Well, certainly what you just talked about, the primers come much faster, right? We've done qPCR before with a reason why a lot of people chose Cyber Green over TaqMan, right? It's not because they only want to look at one target. It's because it comes fast and it's cheap, right? So there's that aspect of it. But one thing I didn't really highlight as a benefit of being in the single molecule realm is that so if you can see my screen, myself here, you can actually think about if you are looking at two targets in two different spaces, you actually are amplifying your targets in isolation. So in the sense of multiplexing, yes, in the same tube, you're multiplexing, but in the compartment, you're actually singleplexing. So what happens in the biochemistry of PCR is that you don't have to worry about this whole assay interference or whether that target has a higher GC content, this one is a longer length, those kind of things. The amount of thought you need to put in is dramatically reduced. So that's kind of why keep emphasizing that optimization is fast, because that's actually an issue with PCR right now, right? Like if you multiplex in qPCR, you add 1, 2, 3, 4 targets, the CT values are different. But in single molecule realm, the counts are basically the same. Sorry, long-winded answer.
Okay, I think it's good to hear sort of the theory behind it. I'm going to cover a few more questions. We've had a couple to this effect. How long is the accrual time for sample, or the actual time, sorry, for sample preparation and reading from start to finish? So kind of walk us through that, setup to imaging and how long it would take to do that.
Yeah, so on how you set up the PCR reaction, I think it's about the similar time of how you would set up a normal PCR reaction, right? And as I kind of alluded to one of the slides, you can process and centrifuge 48 samples per spin in 20 minutes, so that's 20 minutes. And the thermocycler we use is just a standard off-the-shelf 96 well, so you can amplify up to that number. That's typically our PCR amplification protocol is 1 hour. So far that's like just 1.5 hour-ish of time. And then the counting, we count and we image and we count, and the first data comes out within 5 or 6 minutes of the imaging time. So it really, Stephanie, it really depends on how many samples they're imaging at once. If they're imaging one sample, you get it in like 5, 6 minutes. But if you go through 96 samples, then it would be, I would say, 5 or 6 times 96, whatever that number is.
think
Yeah.
it's in the 5 to 6 hour range, but
Yeah.
yeah. Thank you. So we're kind of nearing time. We're already 5 over the half hour. I do want to kind of wrap up. There were several questions kind of asking about the sample workup program or early access program. Unfortunately, I cannot drop a link in chat, which I was hoping to do. But if you are interested in participating in our sample workup program, you can email Aline at isham at countablelabs.com. Or you can also visit just countablelabs.com and reach out to us through there. Also look for a follow-up email in the coming days, which will have more information on it. So if you are interested in either, helping us improve our product or also trying out your own samples or talking through how your samples would work in the Countable system, then Eileen and the rest of our applications team is happy and ready to talk to you about that. So yeah, anything else, Eileen?
No, I hopefully, yeah, I think it went well. Thanks. Thank you for coming, everybody.
Awesome. Thank you all so much. Have a great rest of your day. And yeah, look forward to some follow-up later. Thank you.